So I am here to talk all about Fairchild's South Florida Plant Conservation Program. And thank you for the nice introduction, Brian. And it's nice to be here with all you at the Dade chapter of Florida Native Plant Society. So thanks for inviting me. It's been a couple years since I've spoken to the group. And tonight I have a presentation that's kind of in three parts. So the main three parts are: why does South Florida need a rare plant conservation program? What do we, the conservation team, do? And then I'm going to talk about what we do with three conservation stories that are some illustrations of some of the work we do. So for the first part of this, I'd really like to explain and illustrate how we are in a biodiversity hotspot. And this is something that is not always, we know there's rare plants here, but we're really exceptional in the country and in the globe when you look at the bigger picture. Back when I started working at Fairchild, South Florida was considered part of the Caribbean biodiversity hotspot. There were 35 biodiversity hotspots recognized in the globe, and we were part of that one. And then I think the Conservation International came out with revised list of hotspots, and they made this little dip in their Caribbean hotspot, and they cut South Florida out of it. So for a couple years, we were not in a biodiversity hotspot. But just recently, in 2015, there is a paper published about a new biodiversity hotspot, the 36th one in the world. And this is the North American coastal plain. So we jumped hotspots from the Caribbean to the continental US, but we are once again officially in a global hotspot. And what that means is, being in a hotspot, is there are high levels of endemism of plants and animals, meaning plants and animals that are found in that region and nowhere else in the world. I talk a lot about Miami-Dade County in this presentation, since this is the Dade chapter of the Native Plant Society. So looking just at our county, we have 60 taxa, 60 plant taxa that are found only in Florida. And we've got 14 federally listed species, 12 endangered, 2 threatened. Those are plant species, rather. And then on top of that, we've got 2 endangered butterflies, an endangered bat, and an endangered beetle, the Miami tiger beetle. We really stand out from the rest of the United States, the rest of the continental United States, when you consider our biodiversity. And to illustrate that, I'm going to show a series of maps that are really cool. And many of you, if you haven't seen them before, you're going to wonder where they come from. And it's from this website called bonap.org. This is the Biota of North America program. And it's looking at the continental United States and the biodiversity within. So here's a map showing the native tree species density in the United States. And this is essentially a heat map where they're looking at the number of species that are native to each county. So the highest levels of diversity are pink and light pink. And you can see that there's around like the Tallahassee area, there's a lot of diversity. And then South Florida is very light pink. And if you jump in and look at South Florida, we are actually #1 in the whole continental US for native tree species density, with 154 species. And Collier and Monroe County are a very close second with 152. So this here is a look at the number of native range-limited vascular plant species, which is just kind of a fancy way of saying the number of naturally rare plants. And again, you can see that we in Southeast Florida are up on the high end. We're actually #1 for this measurement too, with 303 native native range limited vascular plant species. And of course, one of my favorites is fern species density. This map is a little bit different, and I honestly don't completely understand how they did the measurements here, but you can see it's not by county, it's more by I think bioregion, and the most dense is a blue color, and you can see a little bitty dot of blue down here at the tip of Florida. And right around where the Miami Rock Ridge is, there's this little blue circle and the number 60. And we are the hot spot in the whole continental US for fern species diversity with 60 native species by this BONAP definition. And then one more, here's a map I pulled out for epiphytic species density. And this is probably not going to be a huge shock to anybody who's looked around Miami that we have really exciting diversity of native epiphytes. And you can see South Florida is light blue, and we're #1 here again. We kind of share the #1 with Collier and Monroe County with, again, 60 species. So all of that is really illustrative, I think, of how special the place we are is in terms of biodiversity. But there's some bad news too. And some of it you can see in these BONAP maps, just like you can see the good news. This is a look at the number of exotic vascular plant species. And Florida lights up here too, South Florida. And I'm kind of surprised we're not #1, but we're #6 with 523 exotic vascular plant species. And then switching sources from BONAP to the Institute for Regional Conservation, we also are home to a large number of species that have been extirpated or may be extirpated. By the IRC's count, that's 62 species that have been wiped out of our county, 11 that are likely extirpated or historical, and then 229 more that are listed as threatened or endangered by the state of Florida. And then this is an example of one of Miami's extirpated species. This is the Florida Keys indigo, formerly in Miami-Dade and Collier, now only found in Monroe County. And even though we're a plant society, it's good to think about all biodiversity and the fact that it's not just plants that have really drastically declined in our region. Butterflies have had a major decline as well. There's been a lot of attention in the literature the last few years about butterfly decline in the central prairie region of the continental US, the tallgrass prairie. But South Florida is another area with extreme butterfly decline. And oops, go away. I've got a pop-up. Remind me later.
Jen, one quick thing.
Sure.
On your screen, do you see on the bottom that it's sharing your screen? Do you see the hide button?
Yes.
Could you press that, please?
Oh, yeah. Thanks.
Sorry about that. Thank you.
Okay. So this information about rare butterflies in South Florida, it comes from a paper that I don't think was officially published, but you can find a PDF on the internet. It's by Mark Minow, Jarrett Daniels, and Dean Jew, and they call it Statement on the Extinction, Decline, and Loss of Butterflies in South Florida. And they talk about our 2 extinct subspecies found here and the 17 more butterfly species that are on their way to becoming extinct if things don't turn around. We've lost birds too. The red cockaded woodpecker and the Florida scrub jay, they both used to be found in Miami, but they both need old growth pine forest, so they can't live here anymore. We've lost quite a bit of mammals. The deer, the panther, the black bear, the bobcat, the fox squirrel, the mink. river otters, although they still show up once in a while in Miami, and 2 species of skunk. They've been pushed out as South Florida has become massively developed. So I know I'm preaching to the choir here, and I probably don't need to convince anybody that there's a critical need for conservation in South Florida, but it's nice to kind of look at these numbers and these range maps of things and just see it kind of slapping you across the face how important our region is for conserving biodiversity. And for the rest of this presentation, I'm going to talk a little bit about what we, Fairchild's conservation team, do to conserve our local plant biodiversity. And this is, you're looking at the conservation team here. I'm the program manager. We have 2 biologists, Lydia Cooney and Jimmy Lang. Sabina is our seed lab manager. Daniela manages the Connect to Protect network, and Brian is our conservation horticulturist. And then I'm afraid I probably forgot somebody, but we work all the time with volunteers, and we're so busy, we have a lot to do, and we couldn't do it without all of this amazing help from these people. So I want to especially acknowledge our volunteers for all the work they've put in with us over the years. So what the South Florida Conservation Team does, we have a program goal, and it's to reduce the extinction risk of the rare flora in South Florida and the Caribbean. We primarily work in South Florida, but we also do a little bit of work with plants that are native to Puerto Rico and the U.S. Virgin Islands, and that's largely because those are U.S. territories. So there are species there that are part of the U.S. Endangered Species Act. We do I'd say 10 to 25% of what we do, depending on the staff person or the time of year, is outreach and networking. We all try to have a presence on social media. We all try to publish, although we never do that as much as we should. We're usually mentoring interns, whether they be from Fairchild's Biotech High School program or from FIU or another university. We give presentations like to the Native Plant Society. And then we run the Connect to Protect network, and that's primarily run by Daniela Champney. That's our outreach program that we started in 2007. And I'm not going to be talking about that in this presentation, but I will put a link at the end because you probably all know about this program, but if not, I really encourage you to join. What I'll be talking about mostly is our behind the scenes work, which is most of what we do. We do a lot of what we call in situ rare plant monitoring, meaning in site or in nature. We maintain nursery collections. We do a lot of seed banking and seed research. We do applied research, especially in conjunction with land managers. We look at a lot of management questions. We do rare plant reintroductions, and those numbers 86 and 25 is to remind me that by my count, we've participated in 86 different reintroduction actions of 25 species since our program began. And of course, what some of us do more than anything is a lot of grant writing and reporting. So I'm going to tell you about 3 conservation stories that I picked out, in part because I think they're kind of fun to talk about. A couple of them are things I haven't really spoken about in a public presentation before, and I thought these were kind of interesting stories that explain kind of the big picture and how we use all of the resources we have at Fairchild to work on plant conservation. So I'll go through these one at a time, starting with the Blodgett's silverbush relocation. So Blodgett's silverbush it goes by the Latin name Argithamnia blodgettii. This is in the Euphorbiaceae family. This was added to the federal threatened species list just a couple years ago. And it's a South Florida endemic. It's found only in Miami-Dade and Monroe counties. It's found in pine rockland, hammock, especially edges and gaps of hammock, and sometimes in the Keys in more coastal habitat. In Miami-Dade, by my count, it's in about 6 conservation areas, so it's pretty rare. And then it's honestly not the most striking plant. You might just think it was Spanish needle walking by it. The flowers are really not showy at all. They're tiny and green. One thing I like about it that I think is kind of cool is when you break some of the foliage, and if you make an herbarium specimen, or if you just knock into a plant and damage it, the damaged foliage turns this cool silvery blue color. So that's what's in that upper right-hand picture. And like a lot of euphorbs, it has a 3-seeded capsule that pops open when the seeds are ripe. So this particular project that I'm talking about has to do with the Owasso Bauer Edition Eel Preserve, which I think many Dade Chapter Native Plant Society members will be familiar with. This eel preserve is really a gem among gems in the eel program. It's a tiny little square of pine rockland that's near Camp Owasso Bauer, so that's why it has its name. It has really high diversity. It's just chock full of native plants, including many, many, many listed species. But a couple years ago, the Chrome Avenue widening project started to creep south more and more, and it was determined that the edges of the eel preserve needed to be impacted in order to widen Chrome Avenue for safety reasons. So there was a lot of back and forth over, I don't know, a year or two between the eel program and the state. And in the end, the road widening project won out. It was decided that the project would move forward, but the state and their contractors had to relocate the Blodgett silverbush that was in the impact zone. They had to be taken out and put somewhere else. And this is because, legally, this is because this is a federally listed species. There are other rare species there too, but they had no legal protection. So Fairchild was subcontracted under one of FDOT's contractors, a company called AECOM. And we were chosen because the Miami-Dade County asked them to choose us for this rescue. And our plan was to collect Blodgett silverbush seeds, which is something that's not normally done in a plant rescue, but we really urged the DOT and AECOM to let us get in there as soon as we could to start collecting seeds for as many months as possible. And they obliged. We rescued plants from the impact zone, and you'll be happy to know that we not only rescued Blodgett silverbush, but a lot of other things too. And then we relocated the silverbush to some other protected sites. So we actually had 6 months of seed collection, and this work was done almost entirely by Brian Harding. He was able to collect 546 seeds in the 6-month period from the edge along Chrome Avenue. And this was despite some adversity. That picture in the middle with the tag is showing one of the plants that got mowed. I think 2 times we had all these no-mow flags, and 2 times some Yahoo accidentally mowed the plants. But this was a real learning experience on how to collect seeds. We started trying to just collect seeds off the plants one at a time. finally figured out that putting these organza bags, this like bridal veil material, over the end of the plant and just letting the seeds fall into that was so much easier. And that's a technique we use a lot now. So we ended up with those 546 seeds. They came to Fairchild to our seed lab. And this picture here is Dr. Savina Wintergurst, our seed lab manager, with last year's interns from the biotech at Richmond High, Melissa and Janae. And what happened with these seeds were most of them got put into long-term storage, and they are still in our freezer at Fairchild. About 20% of them were sown, and we really wanted to sow some plants and get seedlings going because we were being paid to translocate plants, but we weren't sure that we could actually rescue the plants. So somehow we had to do this. So this was kind of our backup policy. And then some of the seeds were used in seed research. We ended up, the seeds that were sown did quite well, and we had a lot of plants growing in our nursery, and we ended up using those for seed bulking. And here's a picture of Janae collecting seeds from those organza bags in our nursery. So we were able to collect hundreds more from the nursery thanks to that seed bulking effort. And then something that's really cool about having our seed lab and a scientist like Sabina in the seed lab is these seeds were really hard to germinate. We knew that they needed something that they weren't getting. I think germination rates were maybe around 10%. So Sabina decided she was going to figure out how to break seed dormancy. And these are all the treatments she looked at. Soaking in water, the potassium nitrate helped a little bit, but boy, putting the seeds through some smoke water really broke the dormancy in these seeds. And you can see her in her pictures when the seeds did not get smoke treatment. She had no germination. I think maybe there's one starting to germinate here, but in the seeds that were treated with smoke water, they just germinated like gangbusters. And the smoke water really was pine needles that she set on fire in a little bee smoker, and she bubbled the smoke through some water and then used that water to water the plants. So the smoke extract triggered germination in this species. And it was very fortunate that we were able to take advantage of this somewhat sad situation and use it to find out a lot of really interesting things about the biology of this species that's going to help us to better conserve it in the future. So finally, we got down to the time for extraction. And in June and August of 2019, we ended up taking out 153 plants. The young lady on the left is my niece, Logan, there. She's taking out a plant with a digging bar. And then we have staff from EEL and our volunteer, Nathan. And Mike Briner from AECOM is kneeling in that picture. And then Rob McMullen from DOT. They were both out there. And they were really excited about this project too and committed to seeing it through to the end. And they were also really fun to work with. So we ended up getting 150 plants extracted. There's Logan with one that she just dug up. And then out of the 153, that picture on the right, that's what they look like when we took them out. We cut off all their leaves so they don't dry out, and pot them up and hope that they recover. And we had 57% survival, which is pretty good. A lot of times I think there's a misconception, oh, you can just pick up plants and move them somewhere else, they'll be fine. But that's really not easy to do, especially in Miami where our soil's so rocky. But it's not something you can count on at all. But we had 87 plants. So we relocated those plants to appropriate habitat, and that in itself was a process. We looked around. In this picture, the star in the center is the Camp Owasso addition. So we looked at appropriate habitat that was within a couple miles of the Owasso addition, and we considered UF's Pine Rockland at Trek, West Biscayne Pineland, Ingram Pineland, Seminole Wayside Preserve, Hattie Bower. Hattie Bower is now a hammock, but it used to be pineland, and Argenthamnia blaggetii has been extirpated there, so that's why that was on the table. And then Owasso Bower, Camp Owasso Bower. We ended up choosing West Biscayne and Seminole Wayside as the most appropriate habitat. And this is a decision we came to with the eel program and the AECOM biologists as well. So the plants had a really short time to recover in the nursery, just over a month. And then we were putting them out back into nature. So we put them into these two pinelands 39 or 43 days later. And after the first 3 months, we had 78% survival at Seminole Wayside, and it was quite a bit lower at West Biscayne. That's in part due because the plants we put at West Biscayne were smaller, but it's mostly due because we had these really jerky little raccoons that lived next to West Biscayne, and they kept digging up the plants and we'd have to go back and replant them. We finally hired somebody to come and check on the plants a couple times a week and replant them for us because it's quite a drive. So we found somebody that lived closer that was able to do that for us. And in the end, at our last check, we had still 78% survival at Seminole Wayside. This guy in this picture here, this Blodgett silverbush, look at how happy that looks. And we had 43% at West Biscayne. So we haven't noticed any seedlings yet, but we're waiting for the day. These plants have been flowering and fruiting prolifically ever since we put them in. And then we had the opportunity to do a little tiny experiment because Miami-Dade County NAM, they decided to use their burn box in part of Seminole Wayside Preserve. This is an aluminum box where they can do a little tiny controlled burn. So they did that, and Sabina and I threw down 3 Blodgett silverbush seeds before they did the fire. And then very recently, which was 1 year after we put the seeds down, Lydia visited the spot to check, and lo and behold, there was a Blodgett silverbush seedling that obviously came up after the fire. So that was really cool. We don't know yet if our reintroduced plants are going to make seedlings, but I have a feeling that if Seminole Wayside or West Biscayne experience a prescribed fire, we're going to have a lot of baby Blodgett silverbush. And instead of having a lot of acknowledgments at the end, I just wanted to kind of acknowledge as I go. And I wanted to mention Brian and Sabina, and especially Miami-Dade Eel, Derm, and PROS, the Parks Department, and FDOT and AECOM for helping with this project. And I know I forgot people, but there was a lot of people that helped to rescue plants on this project. It was a big effort. But now I'll go into story number 2 about what the conservation team does to conserve some of the incredible biodiversity we have here in South Florida. And this is our fern conservation program. And it's hard for me to give a PowerPoint without talking about ferns. I do a lot of work with ferns and I really like them. And this program we're really making an effort to reduce the likelihood of extinction of South Florida's rare native ferns. Currently, we have, I think, 48 taxa that are in Miami that are native. That's the highest of any county anywhere in the continental US. And we didn't really set about to make a fern conservation program, but it just kind of slowly happened over time because so many of the areas where taxa are ferns, and ferns need to be treated differently than other flowering plants in many cases. So what this program does, we go out, we collect wild propagules, almost always, probably 95% of the time from eel properties in the Miami-Dade Eel Program. We propagate we start ex situ collections. We try to store spores long term, and then we reintroduce ferns into appropriate habitat where we can. And by far my favorite one of those objectives is objective 1, to collect the wild propagules, because that leads me to the hammocks of Miami-Dade County, like this picture here from Ross. And it's just a real treat to visit these grottos in Miami-Dade. They're beautiful. Objective number 2 is to propagate. And in the first several years of this program, we didn't do any propagating at Fairchild. It was done primarily at first by the Cincinnati Zoo. They have the Lindner Center for Conservation and Research on Endangered Wildlife in Cincinnati. If you ever in the area, I highly recommend visiting it. They have a really great program for visitors. The zoo itself specializes in small cat conservation, and I'm not talking about house cats. I'm talking about like cute little Arabian sand cats and things like that. But they've also have this side branch where they started working on, I think, in vitro fertilization and things like that with animals, but they started working on plants too, and more they work with rare plants. And that lady in the corner looking through the scope is, I think, Valerie Pence. And Valerie is very good at fern propagation. So she propagated many of our early ferns, and they would just ship them to us, make a little fern burrito like this in wet paper towels, and they'd send it to us, and then we'd finish the job. And then more recently, Mike Friedman, who was on horticulture staff at Fairchild for, I think, maybe 30 years, he was really into fern propagation in the 70s, and then he kind of gave it up. But then when he started working with us more and more and started propagating ferns in-house, and because of Mike, we just really figured out how to do it all at Fairchild, and we do much of it now. So this is a look at our fern propagation area. We do everything in these little deli containers. We have a volunteer named Isabel that comes in once or twice a week to work on this, and she does an amazing job. And we have all these species that we're propagating. And mostly it just takes patience. It's very low tech, kind of tedious, but nothing real complex about it. We've now propagated ferns from 16 genera, and when I was trying to count, I put a little dot by each genus for how many species, so I came up with 26 species. This is a list of everything we've propagated. From all of those ferns we've propagated, we have some great ex situ collections going. We have a lot in Fairchild's nursery. You can see here on the left. And then I don't do it enough, but we are able to collect spores and store them. But they have to be stored cryogenically. They can't be stored in a regular freezer. And Fairchild doesn't have a great cryogenic facility, so we have to work with partners to do that. We tend to ship spores to the USDA National Germplasm Lab in Colorado. But FIU is also a potential for us to collaborate with in the future, because they do have cryogenic storage facilities. But we have plants that are established in the ground too. There's a few of our ferns in the Isabel Foster Fern Glade at Fairchild. These are in that lower left, that's Thelipteris grandis outside of our Gatehouse Museum. And then by far my favorite planting is this one on that big picture on the right. That's at the Deering Estate. And the horticulturist there, Rose Roberts, took plants that we grew, Tectaria, Heracleofolia, the broad halberd fern, the brittle maidenhair, and some strap fern, and she put them in this gorgeous planting that I just love. And this replaced wart fern, so I was very pleased about that. And then we do a lot of fern introductions, and so far we've done 7 species into 3 locations. And I'll just kind of go through the 3 locations. First was Hattie Bauer hammock. And we've put in 56 climbing vine ferns, the Microgramma heterophylla, and we've put in 254 broad halberd ferns. These have done great, and as far as I'm concerned. The climbing vine fern especially is just gorgeous. It's taken off, it's climbing, it's sporulating, they look amazing. 66% survival is really wonderful. The broad halberd fern, we kind of had a strategy of put out a whole bunch and then don't provide as much care. And that seems to have worked okay. There's at least 30% survival. It's really hard to measure survival though, because we put so many out, and then post Hurricane Irma, the hammock was just really trashed. So it's very difficult to relocate the plants we put out. But at any rate, there's at least 75 still out there, and they're already recruiting the next generation of baby ferns. At the Deering Estate, we've put out 3 species. The grid scale maiden hair fern, we've got 32% survival of 211 plants. Some of the surviving plants are really robust, and we've also seen next generation recruitment there already. Very recently, we put in 26 brown hair comb fern, Tinnitus submarginalis. Some of these have been kind of drying out a lot, but we still have 100% survival, and that's largely thanks to Dallas Hazelton and Vanessa Trujillo at Deering, because they did a lot of supplemental watering over our summer, which was supposed to be the wet season. And then we've done modest spleenwort, which this one's been really tricky. We only put 4 individuals out because I don't have very many, and I wasn't willing to put any more out. This species is very delicate, very hard to grow, and we continually have problems with raccoons messing with our stuff. So you can see in this picture, Dallas has a big thing of metal mesh, and he's creating a protective structure to put over the ferns to protect them from raccoons. And we did have 1 fern die, but the other 3 modest spleenworts that we've put out there are doing amazing, and they look great. So hopefully there'll be more to come next year. And then our last fern project, I don't want you to think they all are a success. We've put ferns into 2 eel preserves, Silver Palm Hammock and Silver Palm Groves. And this has not been very successful. I had 162 individuals. They were gorgeous looking, healthy ferns. You can see them in the background on this slide. Those were grown by Crew, and they came to us as fern burritos in the mail. But these plants just did not fare well, and I blame 2 things. One, it probably won't surprise you, raccoons. We had, especially at Silver Palm Groves, we had raccoons just continually digging our plants up. And I noticed that the preserve is right next to an avocado grove, and there was just tons of raccoons living in the grove, eating avocados all day long, and then going out for fun and trashing our fern planting, I think. But the other problem, I think, is we just didn't plant them in the right spot. We chose areas that were too wet, and we're trying again this year in some areas that are fully shaded but more dry in Silver Palm Hammock. So hopefully that one will be more successful, and we still are in the process of doing that one. So in the future, one thing I know that you all, this audience of the Native Plant Society, will be really interested in is I've been working with Teresa Tremansky at Miami-Dade College, and she's really interested in trying to get more ferns out to landscaping as alternatives to some of these nasty invasive ferns that are really the only thing you can buy, like wart fern and tuberous sword fern. So this is going to be a couple years into the future because ferns take so much patience, but we're working on it. We do start ferns for other conservation practitioners. We've been growing stuff for with Withlacoochee Forest up near the Brooksville area, and we have a few ferns from Puerto Rico that we're growing. And we hopefully will be banking spores with more regularity. And I have a lot of fern people to acknowledge here. I think I mentioned most of them already, but I wanted to especially mention Steve Pearson and Joyce Mashinsky for everything they've done in this program, because I hadn't mentioned them yet. And I also have to mention that I have if you like Instagram, I have an Instagram account that's just 100% dedicated to ferns. So if you want to follow me on Instagram, I'm at ferny.sofla for South Florida. All right, so my last story of conservation is long-term monitoring of the U.S.'s only remaining wild population of Sargent's cherry palm, Pseudophoenix sargentii. So Sargent's cherry palm, you're probably all familiar with it because luckily it's not super rare in the nursery trade, although I know it's expensive. It is a slow-growing, long-lived palm found throughout the Caribbean basin, including South Florida. Nowadays, there's only one population left in the wild that's naturally occurring, and that's on Elliott Key. In the past, there were plants on Long Key and Sands Key, but those are now extirpated. And it's not just Florida, many of the Caribbean populations have really been in decline. This picture is my former coworker, Sam Wright, with one of the palms on Elliott Key. This palm has a long, sad history of basically abuse by humans and being an endangered species. This picture is from 1920. J.K. Small took this 100 years ago, and he says this is apparently the only mature specimen left on Elliott Key at the time of the photo. And people were just crazy about these palms for landscaping, and they would harvest them and send them elsewhere in Florida to be planted in estates. And here's a picture again from our Florida archives, the online archives, that Small took in 22. And he says, there are 24 palms remaining in this grove on Long Key, where there were several hundred palms when discovered 40 years ago. So he was lamenting the destruction of this species. And it looks like the one in this picture is probably about to get taken down since it has cables around it. So Fairchild's had a long history with this species. In 1991, our conservation program manager, Carol Lippincott, did a census on Elliott Key and found 47 plants. And she worked with a lot of different partners around the area to propagate plants and do some reintroductions. I think in the pictures here in the upper right, that's Rob Campbell planting one of the juvenile palms, and then I think it's a lady named Ann Deaton watering one of the juvenile palms. But you can see this was a very labor-intensive effort. You had to take, or they took 52 seedlings and 11 juvenile palms by boat to Elliott Key, had to dig holes, had to carry in the water, and the plants had cages around them to try to protect them from herbivory. So over 2 years, they put out 63 plants. We last assessed the population in 2017, and that's 26 years later. So of the 52 seedlings that Carol and Rob put out, only 2 were still alive, but those 2 were quite healthy. And of the 11 juveniles, 3 were still surviving. So a lesson there, if we do this again, is we would probably put our resources towards putting out juveniles if we had that option. In this picture, Jimmy Lang here is standing next to one of the juveniles that was planted in 1991. And you can barely see it in the picture, but there's an old inflorescent stalk on that tree. That tree managed to flower and hopefully fruit. It's not in the greatest shape, but it managed to do its job. So that's the first time we've seen that of the reintroduced plants. The number of wild adults on Elliott Key is not good. Since my time at Fairchild, there were 11 the first time we went out, and when we went out most recently, there were only 6 wild plants. And there's been some reports that this species is extinct on Elliott Key, but it's not, the story is not good, but it's not gone yet. It is true that we've been going out doing monitoring, and every year we find more dead adults. Scenes like this, where a former pretty palm tree has turned into a dead trunk. But the population is not just adults. At our last monitoring, we counted 203 total plants. And when you think about that sad graph of the number of adults going down and down, it's not all bad news. We've been tracking palms over the years and looking at how the different size classes are doing. And you can see that fat orange line is what we call the large juvenile. Those are plants that are almost big enough to produce flowers. And everything's going down, but the large juveniles are going up. And if you have to have, if you have to have just one size class going up, I like the idea of large juveniles going up. These are palms. They look very robust, and in the next few years, they're going to come into adulthood and start flowering and bearing fruit. And we're very hopeful that we'll be documenting a increase in the population again. Jimmy and I went out to the island right before the pandemic. We were lucky enough to get out there with Vanessa McDonough and Shelby Moneyspith. And we spent a couple hours doing a kind of a quick monitoring. And one of the naturally occurring plants, we looked up and saw something red up there and said, "What's that?" And they were making fruits. And there was quite a few fruits in the canopy and on the ground. So the population is there, and it's trying its best to hold on. And hopefully these little guys will turn into some healthy seedlings. And we will be going back. We do monitoring these days every 4 years or so. So our plan is to go back in mid-January. And I want to put this out there. Anybody watching this that has a yacht and a lot of spare time and wants to drive some biologists over to Elliott Key or pick us up, please let me know. The park does accommodate us, and they're going to be helping us to do transportation, but because of COVID restrictions, they can't take very many of us, and it would be nice to be able to take more than a couple people, because this is the island, I think, is 8 miles long, and there's hundreds of plants, so it's a bit of an effort. So I wanted to acknowledge all of these folks for the Sargent's cherry palm work that I presented. I think I mentioned most of these people, but I didn't mention Janice or Joyce, I don't think. And Janice and Joyce have really put a lot of work into this species over the years, especially Janice. Has been a really long-term effort for her. And they have a nice publication about Fairchild's restoration efforts with this species. I can't remember the journal. It might be Restoration Ecology. So that's the end of my stories. And I have a couple more slides just to wrap up. I wanted to, of course, mention some of our current funding sources. FDACS is our biggest one in Miami-Dade and Broward Parks. We have, I think this is, what is that, 10? This is just some of our current funding sources. We have a mishmash of a lot of different support to be able to do what we do. And I also didn't want to not mention that I didn't mention the work done by Lydia Cooney. I specifically left her projects out of this because I know she just gave a presentation to the Native Plant Society about her work. So I thought I'd focus on some of the other stuff that we've been doing lately. But she's been doing a lot of really exciting work with plant restoration, especially in the pine rockland. And I didn't mention the Connect to Protect network either, but Ms. Daniela runs that very capably. And I think if there's any take-home message here, it's that there really is a critical need for plant conservation here where we live in South Florida. And so we're doing everything we can to conserve plants. And I want to thank everybody in the Native Plant Society for doing all you do to encourage the use of appropriate native plants and to do what you can because it's important. So that's it. I thought I'd end on this slide. This is our team website. If anyone has interest in learning more about our publications or our seed lab, there's some good resources here. And then Connect to Protect. This is our homeowner program where we encourage people to grow pine rockland plants in your yard.